Great. Fantastic. I can't see anybody, but I'm imagining a room of important people and scientists, and many of you I know as friends, so great to be presenting to you. And also it's a great honor, and thank you to all of you for continuing to invite me to give some thoughts on things which, you know, to some extent I've been to New Zealand for a while, and I'm also very far away, so I'm sorry I can't be there in person. I did save 6.6 tons of carbon by not coming, so that's not a bad thing. And a word of sympathy also for Auckland. I mean, in the summer of 2021 we had some tremendous floods here, and it's really quite scary how things are changing much faster than I think any of us imagined on the climate change front. So I think it's worth repeating, it's been said before, but I'm going to say it again, is just how successful SHIVERS-I was, not necessarily from the scientific perspective or the public health perspective, which I'll talk a little bit about, but also just the fact that there was this really very vibrant collaboration that was constructed by Sue and her colleagues, which is listed in the affiliations down below on this slide, and also the fact that it spawned all these other really important and really interesting and internationally recognized projects on influenza epidemiology and immunology and a variety of and virology, which have really been important contributions. So, where's it? Let me get to my next slide. Do it. Excuse me. Here we go. So, just going back a little bit as to what was the purpose of SHIVERS-I when we put out the call for proposal. And really it was born out of the 2009 pandemic, and there was this feeling in April 2009 that, you know, with the pandemic appeared in Mexico, and then everyone was kind of on tenterhooks, what's going to happen in the Southern Hemisphere winter. So we monitored the winter, and then we realized that the pandemic wasn't over, so we wanted to be able to be sure, not only for the 2009 pandemic, but also for other future pandemics, and for seasonal flu, to be able to have a better understanding of pandemic and seasonal influenza in the Southern Hemisphere. On top of that, there was lots of need, there was additional data suggesting that, at least in 2009 pandemic, that the indigenous groups were at least 5 times more likely to die of H1N1, even once adjusted for underlying conditions. And this is something Paul Thomas, I don't know if he's online yet, but was interested in looking at genetic factors and predisposition issues. So that was also an interesting aspect of the proposal, and also a bunch of different data that I think we still needed to get, and still need to get, you know, on elements of seasonal influenza which are still poorly understood. So when we wrote this thing, we wrote these 9 objectives, and Sue, I think, was, when we write these cooperative agreements, we generally throw everything in there, because this is, we try to make it as broad as possible from the US government perspective, because it allows us to have flexibility depending on who's applying, that they may have focus on one or the other, and it would give us some flexibility in how to best work with our partners. Sue, in classical Sue fashion, took this as verbatim, or at least as an absolute requirement, and tirelessly worked on all 9 objectives, just about. I think not just about, I think all 9 objectives have been met by SHIVERS-I, and it was always a source of amazement to me that she had the energy to tackle all of those. But these are the 9, so you can, I think Sue presented them, but you have understanding SARI, understanding VE, a lot of work done with that, and looking at the interactions between influenza and respiratory pathogens, perhaps a little less done on that. Mortality, you know, Michael Baker was working on that, non-severe respiratory illness, the SERA survey work, the risk factors, and I'll come on to some of the data that came out of that, the immune responses, and the economic burden, which I think was also looked at least for RSV. And the platforms, I think Sue has presented, that were gradually, incrementally introduced, starting with SARI in 2012, then the GP platforms for ILI, and then importantly, the longitudinal community platforms for serology and mild asymptomatic infection. And then also what I think is really key in Auckland, which always was exciting, is to have administrative data sets that when you have a relatively robust and comprehensive kind of national health system with little if I remember, little private care, that really gives you a tremendous advantage when you want to leverage the data that you have from your surveillance and from the laboratory testing to be able to leverage to larger data sets. And this is some of the results that have been coming. I mean, I've lost count of the number of papers, but there were the vaccine effectiveness with test negative design, lots of immunology work, both with cell-mediated, and we, Sue discovered the usefulness of neuraminidase serology, risk factors showing that the Maori-Pacific people, low socioeconomic status and people with chronic diseases all had increased risk. But importantly, it wasn't just had increased risk, but we were able to quantify that a little bit, which was something that hadn't really been done before, to actually put a risk factor to it. Expanding, I think, really important, it was always a pet project of mine, is to expand surveillance to be able to understand the overall burden. Young children who may not have traditional flu symptoms, just show us febrile, do they have flu? Is that caused by flu? People who come in and maybe have strokes or non-SARI presentation, is that caused by flu? So trying to tackle some of those difficult issues. Of course, the data from New Zealand was very important for some of the global estimates on influenza burden. In fact, when we wrote the global burden for 2009 pandemic, the paper in Lancet ID, there were only 5 estimates from around the world that gave us the case fatality risk, and one of those estimates was from New Zealand. And then looking at RSV disease, as Sue mentioned, looking at other viruses important, and also looking at adjusting surveillance definitions, working with surveillance as a mechanism, as a tool, and seeing how it can be further adjusted, and looking at things like radiology. Cameron Grant looked at X-rays and using algorithms to sort of understand if that could help us kind of look at influenza. So what I think is really exciting here is this pyramid of being able to, in the same population, being able to look at different levels of infection, and that really is really quite unique. And of course, this was one of the reasons that I think the WHO was in contact with Sue about seeing how this could be a model for looking at global SARI surveillance around the world. And it comes back, I'll accelerate, I'm probably running out of time, but it comes back to this idea of that. I was in Mexico in April 2009, and in Mexico, the table 2 that I present here shows that the early data came out of Mexico in April, showed that we had 24 lab-confirmed cases of H1N1, and 7 of those died. So nobody knew what was going on. I mean, is this thing going to kill half the world's population, or is it a biased surveillance? And of course, it was biased surveillance because they only had SARI. And so it was very difficult in the early days of the pandemic to really understand what was going on. And so my boss at the time, Larry Anderson, said, what you're going to try to do is try to go somewhere where you've got really good quality data. Don't go about quantity, don't go about national data, try to go for really good quality data. And so we went to a town called San Luis Potosí, where they were looking at all the deaths, all the SARI cases, all the medically attended cases, and they were measuring all the percent of cases that were tested, percent positive. They had a specific catchment area, and out of that came this idea of a sentinel city. If you have all these elements in one place, you can really start to get a handle on some of these pathogens. And I think that's really what New Zealand, in terms of surveillance strength, I think we'll be discussing this a little bit after in the panel discussion, but this idea that you have multiple very high quality platforms at different levels of severity, and of course you have longitudinal data is key, that you can compare one year to another, that you can understand the background burden of SARI. Even if you don't do laboratory testing, you can actually notice differences in clinical picture, age distribution, that can actually tell you things that if it's systematically collected without bias, then you're in a much stronger position to notice public health signals. You have the island advantage, of course, which has been shown with the recent work that we've been doing with Sue on trying to understand the epidemiology of respiratory viruses when they were introduced, and I think there's some really interesting insights for the world on that. And you can get these rapid generation of different hard-to-find measures, and repeating here that New Zealand was only one of 5 countries that contributed this particular estimate to the only burden paper on pandemic H1 that's out there. And potentially also you have a platform for rapid assessment of interventions. So I don't know whether I probably run out of time, but these are a few thoughts about New Zealand could go from here. I don't know whether I should go through these or keep them for, maybe I'll just go through them quickly. I think from my perspective, I'll be maintaining a high quality, systematic SARI and GP surveillance with this denominator data, looking at outcome data, not just logging people with SARI and then forgetting about them, but actually seeing what happens to them. It doesn't happen everywhere at all. Strong laboratory, it's one of the few sites in the world that has this quality over quantity, and has tremendous importance, I think, globally. And even though the idea of seasonality, that the New Zealand winter was going to give us insights for the north, it didn't really happen for COVID, which at least in the last 2 or 3 years hasn't really been particularly seasonal. So that model didn't work. But other novel viruses are seasonal. So H7N9 in China is pretty seasonal. So the fact that COVID wasn't doesn't mean that the next pandemic won't be seasonal. And of course, there are new pathogens such as RSV that will soon be vaccine preventable, and I think it will be really key to measure, and New Zealand could be at the front of that, to measure the impact of RSV vaccination in populations. So maybe I'll just stop there, and then the rest of the points I can cover in the discussion. Thank you very much.